RNA splicing is when precursor or pre-mRNA is edited into a mature RNA. I like to make the analogy of when you were in your preteens and hung out with the people that your parents didn't like, and you didn't really understand why, so your parents removed these people from your life. And when you matured just a little bit, you understood why your parents did this, and that the editing process in your life was too just as essential as RNA splicing. But your parents didn't just remove those people from your life with a blink of an eye, did they? No. It took time and it was a process, and I'm sure they might have had a little help. Well, in RNA splicing, there is some help too. This help comes from the RNA spliceosome. This is the machine that carries out the splicing. These are the parts that the machine is made out of. First, we have an A branch site that is labeled by a single yellow strand. Next, the RNA ligase represented by a rainbow strand. The introns represented by a blue and green strand. A GU region or 5' splice site represented by a red then orange strand. An AG region or 3' splice site represented by an orange then red strand. The subunits to the spliceosome are represented by different color arrowhead bites. Exons are represented by Twizzlers. Here we have a strand of pre-mRNA. In the pre-mRNA, we have exons, introns. At the beginning of the intron, we have a GU region, or otherwise known as our 5' splice site. Move a little further down and you have an A branch site, which is important for the making of the lariat structure that we will cover later. Lastly, we have a pyrimidine-rich region ended by an AG, or otherwise known as the 3' splice site. Our spliceosome is made up of 5 different subparts, with the names U1, U2, U4, U5, and U6. U1 comes and binds to the 5' splice site. U2 then comes and binds to the A branch site. Then a trio of U4, U5, and U6 subparts come and bind to the 3' splice site to complete the assembly of the spliceosome. The 5' end is cut and positioned to be added to the A branch site to start the form and shape of the lariat. U1 and U4 are then released, and U5 and U6 shift positions to better form for their function. The 3' end is cut by the trio and then shifted to complete the lariat shape. The exons are then connected by RNA ligase, and the lariat is released and eventually degraded. The now mature mRNA can be translated into proteins and then can eventually be turned into amino acids. The introns were removed just like the bad influence your parents got rid of. If only mom and dad could have hired their own personal spliceosome, you could have matured quicker.